Our third speaker in this semester's EvoS seminar series. A couple quick housekeeping things before we get started. I do want to say that there will be a reception immediately following. That will be in the terrace. And I think it's accurate to say that we have more food than we've had the past few. So, yes, that warrants applause. So if you want to, if you haven't shown up yet for one of these and you want to see what an awesome EvoS cake looks like, we have one waiting. And second, just a little shout out related to our campus. I know the campus has had this major 3D printing initiative, and I made Steve from Minecraft today, and he glows in the dark. So if you see these opportunities to benefit from that innovation center that they've created on campus, it could be used in lots of different fields, and it's something that the campus is supporting and trying to promote. Students and faculty, look how cool that is. So there's lots of great opportunities there. So I'm very pleased to be introducing Ken Nystrom from the Anthropology department. He has been a member of the EvoS board since the board has started, I think since before the board started. He joined our faculty in 2006. The program started in 2007, so he was on the ground floor. And we're very fortunate to have him as a faculty member here at New Paltz for lots of reasons. He has overseen an extraordinary amount of student collaborative research related to his research in biological anthropology, and I know lots of students of his have benefited from his work. He is currently an associate professor in the Department of Anthropology. He is the lone biological anthropologist on campus, and so we are very fortunate to have a biological anthropologist. He also is the associate director for the EvoS program. And he also holds the position as the Provost Fellow. So he's got lots of cool titles, and it's because he does lots of cool stuff. He has been very productive in the domain of scholarship, lots of things looking at biological remains and skulls, and skeletal remains of different peoples, and he has been awarded the Liberal Arts and Sciences Research Excellence Award for this work, and he has also received a Provost Research Grant for his work. And he's got lots of great publications and presentations to his record. For our program, he is one of the primary people who teaches human evolution. And what I find is when students take Ken's human evolution class, they come up to me and they say, "This stuff's really cool, I want to learn more." And so we're very lucky to have him come today speak about human evolution and about the evolution of human diet. Please join me in welcoming Dr. Ken Nystrom.
Thank you. Appreciate everybody coming out tonight and to listen to a talk that really encapsulates what I teach. A little background for those of you that don't know what biological anthropology is, and more specifically bioarchaeology, but it is anthropology that looks at the remains of life in order to reconstruct some of the behavior. So I look at skeletal material or mummified soft tissue in order to reconstruct how we behave, how we adapt to our environment, to that environment by being either cultural or environmental. And biological anthropology covers a range from everything from primate evolution to paleoanthropology and fossil hominins to prehistoric and historic skeletal collections to modern living groups. So it's a huge field with a lot of different research going on. I am a biological anthropologist, and I specialize predominantly on relatively more recent modern humans. So the oldest thing that I've ever looked at or studied is something about 2,000, 2,500 years old. But also as recent as something that's 150 years old. And in forensic context, in modern settings. So the key is that we can use the skeleton, we can use the dentition to reconstruct aspects of life, to reconstruct aspects in terms of how humans adapt to their environment. So this class is really, I teach a class called the Bioarchaeology of Food. And so this lecture is a smushing together of 15 weeks of information and content into approximately a 50-minute talk. So there's all kinds of information out there on diet for paleohominins, for modern groups, for skeletal collections. And so the point of today's talk is to address a few key points. Why do we want to reconstruct diet? What are the key features? What are the questions that we're trying to get at? Second point is, the main point is really we're going to talk about the methods that we use to reconstruct diet. How do we get at this information? What's the level of information? What's the level of detail that we can get out of skeletal material? Thirdly, we're going to look at the dietary reconstruction of the fossil hominins, the key, really. When we are a hominin, so we're going to talk about some of the earliest examples that belong to our immediate family lineage that we call hominids. And these are basically, and we still are, bipedal apes. And so how can we characterize the differences in diet between these various bipedal apes? And then lastly, we'll talk about the biological consequences of the shift from food foraging to food production. And most of that talk will be taken up with looking at some of or that portion of the talk will be taken up by looking at some of the health consequences. Okay? So that's where we're going. The first question, this is how I started out the semester in my other class, and I asked the students, well, why did you want to take a class called Bioarchaeology of Food? And I had one person respond by saying, well, I'm hoping that you're going to tell me what I should eat. And I said, well, I can't. First of all, you don't know what my diet is like. Okay? And until more recently, a fair combination of this type of material, the McRib, though I prefer some of the KFC stuff instead, with the cheese and the bacon and the deep fried chicken. And the Coke, okay? So I am not here to advocate for a particular diet, okay? And part of that is because there is no particular diet that characterizes or is best for any particular group of individuals. It's impossible to have a broad brushstroke that says this is the diet that you should be eating. Because one of the features of humans is that we have an incredibly plastic ability. We can respond behaviorally, physiologically, we can adapt very well to a very wide range of environmental circumstances and into a very wide range of diet. And we'll look very briefly at that in another couple of slides. So a lot of people were taking the class because they're interested in present-day eating habits. Food is a constant thing on our minds, okay? And in our society, the modern Western diet leads to fairly high rates of obesity, okay? Talking about the index calculated as weight in kilograms divided by height in meters squared. And it has obesity has a lot of health consequences that are well established. And in terms of the United States data, there's a large percentage, though I've heard recently that there's been some changes in some of the younger age brackets in kids. Very young kids are actually seeing a significant decrease in obesity rates. But overall, the United States and the westernized diet leads to high rates of obesity. And so this, at least in part, has fostered some of the more, as my abstract for this talk indicated, has fostered some of the more popular attempts. There's a lot of popular media attention on diet, okay? If you want to make a lot of money, okay, you can come up with a diet, and it will sell very well no matter how good, no matter how bad it may be. But right now, there's a particular movement, there's a particular trend where books are addressing or attempting to address some kind of paleo diet. So the Paleo Solution by Rob Wolf, who came and talked here actually. The Paleo Diet by Lauren Cordain, a couple other manifestations of it. The Paleolithic diet, what it is and why it works, the Primal Blueprint, the Paleolithic prescription. This is a rebuttal to some of the concepts that are advanced in those books. And so this is very much on, every time you open up MSNBC or any of the other news items, there's a good chance that there's going to be something on diet in there. And so trying to teach this class, one of the things is to try to get students to think critically about the information that they're receiving. And so when you see something in the newspaper, if you see something online, if you read a book about the paleo diet or anything else, the point is that I teach you the methodologies and how we actually reconstruct diet so that you can evaluate the claims that these books are making, that these news articles might be making. Okay, so I'm trying to give you the tools. The second big reason why to teach the class really is because food is intimately linked to social organization. Okay? This little diagram right here depicts, this is a classic figure from a general anthropology textbook, where three different levels of social structure. Infrastructure is the mode of subsistence, or food foraging, or food producing. Social structure, so the arrangement of people, the social arrangement of peoples, and then superstructure, or worldview. And you notice that all are connected by arrows. They're all intimately associated. So you will find particular types of social organization in food foraging groups that are different from the types of social organization you see in food producing groups. You'll have different gods and goddesses in food foraging groups versus food producing groups. So there's a very intimate connection between all these different levels of society. And additionally, we have, as I mentioned already, a very wide range of diets. So everything from almost pure animal protein in Inuit groups, though they do have some access to vegetable material, plant food, though it may not be in a particular form that a westernized diet may appreciate, okay, which we can come to later when we talk about Neanderthals, to a predominantly vegetarian diet. And we have the full range probably in here, where we have vegans and vegetarians, and we have raw foodists, and we have people that are more carnivorous and omnivorous. We have an incredible range, incredible plastic ability to respond to, or create our own environment, really. So it's intimately connected to who and what we are. Lastly, species trophic level and feeding adaptations have a strong effect on body size, locomotion, life history strategies, geographic range, habitat choice, and social behavior. So it's really key, understanding diet is key to understanding the ecology and evolution of hominins, of us. Very, very, very necessary in order for paleo environmental reconstruction places these hominins within their context. What diet is available, what foods are available, and what are we choosing from those contexts. So that's the first part. The second part is talking about the methodologies. And I'm going to hit some of these more than others. Some I will barely touch. But there are three basic general categories of data and methods that we could use to reconstruct diet. The first category is genetic adaptations. These reflect, when you hear we are adapted to, or this animal is adapted to eat this type of diet, this is probably generally what they're talking about. Okay. Genetic signals, long-term phylogenetic evolutionary relationships that are manifested in differences in tooth morphology, enamel thickness, or skeletal morphology. Behavioral changes. These are choices made by the individual in terms of what they actually eat. This, genetic adaptations, reflects what they could eat, what an organism could eat, what they are adapted to eat, whereas these data, dental microwear and stable isotopes, reflect what this thing actually put in its mouth, chewed, and swallowed. So this is indirect evidence right here, and this is direct evidence of diet. And you can look at as many teeth as you want, but it's still not going to be an actual indicator of what that individual munched on. The other stuff is a collection of archaeological material, faunal assemblages, so what animal bones are in the ground in an archaeological site. Stone tools. For a while there, when some protein assays were being developed, they thought they were starting to find blood on some of the stone tools. And so we're finding this Acheulean hand axe with blood on it. What type of animal were they butchering? It turns out it was all contamination. It wasn't actually blood, but nonetheless, we can still look at stone tools, and through actualistic or experimental archaeology, you can try to evaluate what these stone tools were made for. Okay? And archaeologists do this. Some archaeologists do this, where they will nap stone tools. They'll find a local zoo who maybe had some animals die, and they will ask for them, and they will butcher them and see how well this Acheulean hand axe can be used to butcher an elephant. What kind of cut marks does it leave on the bone when you're butchering an animal? And does that reflect what we see in the archaeological record? So performing actualistic studies. The last one, other archaeological material. This could be macro or micro botanical, so plant remains, or you can do residue analysis, where if you don't wash your pots well enough at night, okay, you have just a little bit of food stuck on there still, you can sample that, and you can run analyses on that. You can identify pollen, you can identify lipids, you can identify proteins that may still be in that crusted on grime that's on your pot, okay? So we have all kinds of different methodologies, all kinds of different tools for trying to get at diet. Some more direct, some less direct. Okay? We're going to start with genetic adaptations. Those things that are, maybe conceive of them as being broader scale, longer scale in terms of their evolutionary significance. I just put this a picture up here because it's pretty awesome. Though that's an adult male mandrill, and this is not a carnivorous organism. Okay, it is an herbivore. It grazes. It eats grass. So these teeth, those massive canines, right there, are a manifestation of more than likely of sexual selection. They have nothing to do with eating. They have to do with signaling dominance to other males within the troop. Right? But we can look at tooth morphology or tooth shape, and this can lead to some conclusions regarding what those teeth may be better suited for in terms of mastication. The whole evolution of teeth, the appearance of teeth within vertebrate evolution is fascinating. Right? It's really, really cool because it goes along with the evolution of homeothermy, of warm-bloodedness. In order to maintain our warm-bloodedness, we had to chew different. We actually had to chew. And we had to increase our metabolism. So our mammalian ancestry in some way, shape, or form is connected to the evolution of teeth, of mineralized hard tissue. So it's really fascinating background to this. But anyway, posterior tooth size. Okay, generally, the larger the working surface in posterior teeth, the better they are at processing lower quality foods. Makes sense, right? Larger grinding surface, larger teeth, better for processing large amounts of lower quality food. Incisors, I should say posterior teeth are your molars and your premolars. Okay. Incisors are these ones up here. Okay. The larger incisors that one has, generally you will find a correlation in that animal's diet in terms of accessing or utilizing larger or tougher fruits. So you'll see this in chimpanzees. Chimpanzees have very big spatulate-like incisors, and they are predominantly frugivorous. They eat fruit. And so a food that requires a lot of incisal preparation is where you'll find larger incisors in those type of organisms. Smaller teeth is just the reverse, less extensive incisal preparation. Sounds fairly boring, but it is less extensive incisal preparation. So leaves or berries, you're not going to have to bite into a leaf in the same kind of way as you do a chunk of fruit or raw fruit. Shearing quotient is a measure of tooth shape. So we did size, now we have shape. And shearing quotient is really a measure of how cuspy your teeth are. Okay, how flat or how blade-like your cusps are. So on the top part here, the color part, we have a variety of different types of teeth. These would have a higher shearing quotient, how much crest there is on each tooth. Whereas on the bottom, I can't exactly remember who that is, but it's a hominin. Okay, it's not us. But I assume that you can see the difference between the shearing quotient on top, which are a high shearing quotient, and the teeth on the bottom. These are better or more well suited for a particular type of diet than these things are. And so we'll see differences in the type of tooth or the tooth morphology, and that'll correlate with the type of diet. More than likely, this is associated with, you'll find some of these teeth in like insectivores, where they have to bite through chitin, which is a tough material, and also with leaf eaters or foliivores, that they have to shear through that material. And they act very much like scissors. They will shear through the material. These you will find in organisms that are more folivorous as well, with a tougher diet, and so grinding lower quality material. Enamel thickness is another genetic signal of diet. And generally, the thicker the enamel, the better or the more well-suited that particular tooth may be for a hard food. And we're going to get to some definitions here in just a sec. Hard food and also foods that have a lot of abrasives in them, either exogenous or endogenous abrasives. So most plant food has endogenous grit in it. They're called phytoliths. It's precipitation of a silica inside the cell wall. So it's an itty-bitty teeny tiny bit of quartz that's in the food naturally, and when you bite on it, that gets ground into your teeth, and it can leave an impression. Exogenous grit is like dirt, right? And so there's always a little bit of dirt in your food. I think probably the FDA allows a certain amount of dirt in your food, just like they allow a certain number of insects in your food. Sorry. All right. Here is a depiction of thickness among the great apes, enamel thickness in the great apes. So the red is thick, the bluish is thin enamel. And you can see the humans right here. Here's orangutan, gorilla. This is the gibbon. That's the bonobo. And this is the common chimp. And when you generally think of chimps, that's probably the one you're thinking about. And so you can see we actually have quite a bit of, we have thicker enamel than most of the other great apes. But it's also differentially distributed. We'll see this again when we talk about Paranthropus. Paranthropus has thicker enamel than us, but it's distributed more along the sides, whereas ours, we have thicker enamel right on the cusps. Now, they don't really know what this means yet. And they're still doing studies, they're still researching this, the mechanical significance of this differential distribution. But it more than likely is related somehow to mechanical, occlusal mechanics, how these things are coming together to produce bite force and to fracture the food that we put into our mouths. Skeletal morphology. And this is a large area within paleoanthropology for fossil hominids. But looking at how the bones respond to stress. And this is, again, in a longer-term evolutionary scale. So this is a mandible from one of the paranthropines. I think that's a paranthropine. That's a paranthropine for sure. The only reason I think that might not be a paranthropine is because it's not quite big enough. It's not thick enough. Our jaw does not look like that. I should have put a comparative up there, but hopefully you can tell that your skull doesn't look like that, right? If you feel up here, there shouldn't be a large bony ridge, and your face shouldn't jut out that much. You may have very nice cheekbones, but they're not quite as nice as this guy's. Right? All these, the flaring zygomatics, the facial prognatism, this is a sagittal crest. These are all thought to be manifestations of accommodation for very heavy chewing forces. You have all the same chewing muscles that this organism did. Okay? If you clench your teeth, you can feel your temporalis muscles flexing. It's the same muscles in this organism goes underneath the zygomatics and would attach to here. So very large, it would have bulging sides of their head, and these would all be muscle. You have the same exact thing. Same distribution of muscles. It's just that our chewing mechanics have changed because our diet has changed. So this leads to a relatively specific interpretation of what this organism's diet was. They also had, like I said, extremely large teeth. We can also look at the skeleton as a proxy for diet. This is a chimpanzee. This is an Ardipithecus afarensis, one of our about 4 million year old ancestor, and this is us. And what we're looking at is the shape of the thorax, the shape of the chest. And this is a proxy for how big the gut is, okay? We, as humans, have a relatively small gut. And the length of the gut is a reflection of the quality of the diet. So the smaller gut you have, the higher quality diet that you generally have. The bigger gut you have, generally means that you're going to be processing a lot of poor quality material, a lot of poor quality diet. So remember that gorilla? In one of the first slides, I was sitting down nibbling on a branch. They have a big belly, okay? And it's because they have a big gut, and they're processing a lot of a lot of low-quality food, okay? We, and chimpanzees have the same basic manifestation. They have a larger gut, where they have this flaring thorax that implies the existence of a very big gut. And Ardipithecus afarensis kind of has the same morphology. So we can interpret that this individual's diet was more like chimpanzees than it was like us. We have a higher quality diet. Chimpanzees are frugivore slash foliovore, a little bit of carnivory thrown in just for good measure. So we can start making some interpretations based upon the skeletal data as well. The important point here is that all this data, all this biomechanical, all this skeletal morphological data does not necessarily equal diet. It reflects what they could eat. Okay? It reflects what they are adapted for, potentially, but it does not necessarily indicate exactly what this organism was putting in its mouth, chewing on, and then swallowing. And that's what this data does, the next set of data. The non-genetic, behavioral data. The first type of data that we'll talk about is dental microwear, and I admit there's possibly nothing less interesting than talking about scratches on teeth. And I spent about 2 or 3 weeks on this in my class. And I'm sure by the end of that class, all the students are fascinated and thrilled by teeth. Scratches on teeth. But the teeth, the wealth of information that you can get out of teeth is just amazing. Okay? It really is phenomenal how much information you can get out of something just looking at its teeth. Dental microwear are microscopic striations or pits that are gouged into the surface of your enamel based upon what you put into your mouth. It is a temporary signal. It's called the Last Supper effect because the next meal that you eat will start rubbing away the scratches and pits that you just put on it. Okay? We're talking extremely small. Okay, so don't worry. You're not going to lose all your enamel. You could, depending on your diet, but you're not going to. So it is capable of distinguishing between broad categories of food items, and we'll run through that in a minute. But the nature of the dental wear features, the microwear features, there are two basic ones. There's pits and there's scratches. Okay? Very technical terms. Okay? The nature of those things is dependent upon abrasives, again, exogenous or endogenous abrasives, food fracture mechanics, and then occlusal mechanics. So abrasives make sense, right? Okay. Whatever is included in your food. Food fracture mechanics is interesting. It deals with material science, the nature of physical things and how they fracture under stress, how they strain and how they stress, how they morph in shape with the application of force. Okay. So we'll talk, there are two basic types of food from this perspective. There's hard food and there's tough food. Okay. Let that sink in for just a second. Hard food and tough food. Hard food fractures with minimal strain in shape or minimal strain in morphology. So think about hard candy, okay? Your Werther's Original, your butterscotch candy, that's a hard food. Popcorn seeds, any kind of seed is generally a hard food. It fractures before it changes significantly in shape. You have a picture in your head? Okay. This is different than tough food. I always think of taffy. Saltwater taffy is a tough food. It can change shape a lot before it snaps, before it fractures. Okay. Beef jerky would be a good example of tough food as well. Okay. Something that doesn't break with changes in shape until you apply a lot of force. Okay. So nuts and seeds are good examples of hard food. And what happens is that when those teeth come together on those abrasives that are naturally in that food, it presses those abrasives into the enamel. And it makes pits. The tough food, which also has the abrasives in it, you generally find actually the shape of the tooth in this instance tend to be the more cuspy ones, okay, the sharper ones. And so the abrasives slide past the enamel, and it leaves scratches. Okay. We'll look at two examples, two known examples, known diet individuals. This is the black-mantled howler monkey. Right? It is folivorous for the most part. 50 to 75% of its diet consists of leaves. The other part probably consists of small invertebrates, some animal protein, probably some fruit, because they're not going to pass up ripe fruit if they have the opportunity. But for the most part, they are a leaf-eating monkey. The cebus, this little guy over here with the wild hairdo, is omnivorous. Okay, much more of an omnivorous individual. It eats nuts, fruit, seeds. Cebus monkeys are known to pick up rocks and crush nuts in order to get to the meat inside. So they're much more omnivorous. And this is what their dental microwear patterns look like. This is very much what we would expect to see with a leaf eater, where the abrasives that are in the leaves are getting scraped along the cusps and leaving tracks, leaving striations. This is what we would expect from, these are pits that we would expect from the abrasives being pushed into the enamel. This is the old way that this data was looked at, and it wasn't as quantifiable. It relied upon the differentiation between a pit and a striation being longer than it is wide, and a pit being wider. Right? So it was there was a little bit of subjectivity, investigator subjectivity involved. But then they started doing three-dimensional topography, essentially. Okay? And this is the same tooth, okay? But this is a three-dimensional representation of that same two-dimensional picture here. And what it does, this captures the third dimension. Okay? It allows the investigator to look at the depth of these things as well and compare the differences between the two. And we can plot, the two main aspects of this data that we're most interested in are called complexity, which is a measure of how different the image may be. Do we see a lot of striations and a lot of pits, which would be a complex image, or do we see the predominance of one type of microwear feature? So just striations would be less complex. Pits are kind of inherently complex, more complex. And then we have this thing called anisotropy or epsilar, which is a measure of orientation. So this thing over here is from cebus. It has you don't see any orientation to the features there, do you? It just looks like pits. There's no trajectory to it. Here, you can see that everything, all those striations are fairly well aligned. And so they have what's called a high epsilar value, a low complexity and high epsilar. And we would plot that. Here's the ulata, the howler monkey, and they have, this characterizes a tough food, all right, like leaves or meat. These are harder foods over on this end, low complexity or low orientation and higher complexity, more pits, or a combination there of pits and scratches. And so we have cebus showing up over here. When we start interpreting these kind of graphs, we have to not only kind of characterize the general location of these organisms, but we can also look at the variability. If you look at cebus, there's quite a bit of variability on the epsilar value, which means that there's monkeys eating different things in that particular group. Okay? So we cannot characterize an entire troop of monkeys or an entire species of monkeys into a single category of diet. We have to accommodate for individual variation. Any questions about that? Because eventually we're going to talk about, I'm just going to be summarizing these data for the hominids. The next piece of direct data, and dental microwear's direct data tells us exactly what they were putting in their mouths and chewing. The next type of data is biogeochemistry, which is looking at the chemical nature of the food that you eat, and based upon metabolism and breaking down of that material, it is then incorporated into your biological structure. And so the adage, you are what you eat, actually applies in this particular instance. Bioarchaeologists can get at what you eat by looking at the chemical signatures of your bones. The quote that I put down here, I believe I gave the evolutionary studies program, the seminar classes, this article, but dietary signals derived from microwear and stable isotope chemistry are sometimes at odds with inferences from biomechanical approaches, a potentially disquieting conundrum that is particularly evident for several hominin species. This gets back to that one bit that I said the biomechanical, morphological does not necessarily equal diet. So we have some sources of data saying one thing about diet. We have other sources of data that are saying sometimes completely opposite about what the nature of the diet is. This is direct evidence though, so this says actually what they incorporate into their body and digest it, and then incorporate it into their own physiology. There are two principal elements that are used in stable isotopic dietary reconstruction: carbon and nitrogen. Carbon can provide information on the type, a broad category of type of plant food that you're eating. And we'll talk a little bit more about this, but C3 or C4 plant. Nitrogen provides data on trophic level, but also the inclusion, potentially the inclusion of marine food. And I'll show you that in just a minute. So there's two principal elements that the stable isotopes of those elements are used to reconstruct diet. And we can use those isotopes, we can use the elements, isotopes of those elements because of the nature, the unique nature of bone, of mineralized tissue. Bone, I'm a little prejudiced, okay, but I think bone is the coolest tissue and organ in the human body. It is a dynamic, living thing, okay? We generally don't probably think about bone a lot, but it is a dynamic, living thing, and it does respond to environmental stimuli. Okay? It is composed of both organic material, a variety of organic material. The most ubiquitous, the biggest component, organic component of bone is a form of collagen. That collagen is a protein. The proteins are composed of amino acids that are made up of carbon, nitrogen, oxygens, and hydrogens. When we reconstruct diet from the collagen phase of bone, we can take a bone, provided it still has some organic in it, we can take a bone and we can extract the collagen out of it. And we can do a stable isotopic analysis, run through a mass spec, and we can get the number of isotopes for carbon. What that reflects is the protein component of the diet. So when you eat protein, that protein is broken down, and those components are then preferentially redirected to your own protein synthesis because it's ready-made. It's stuff that makes up protein. So your body takes that and directs it towards making more protein, making collagen. The other phase of bone is apatite. It's an inorganic carbon phosphate crystal. This gives rigidity and strength to your bones. This gives a bit of elasticity and the ability to respond to environmental stress. When we reconstruct diet just from the inorganic phase, it's a whole diet kind of signal. Okay? Lipids, carbohydrates, and protein. So it's kind of a mix. Okay? Your whole diet is reflected in your appetite crystals. Because the components of this, so you have oxygen, hydrogen, and this can be sometimes swapped out for a carbonate, and so we get carbon back into there as well. But the big thing to remember is that in some, depending upon the age of the organism that you're working with or age of the material that you're working with, you may not have any collagen left. Collagen really only survives for about 100,000 years. And so all the fossil material that we're going to really be talking about, it's just based upon this. We can't get at what the source of protein was. What that means is that we can't get at whether or not they ate meat. Okay? This allows us to get at if they eat meat, essentially. Okay? But there's no collagen left in the bones when it's that old. So we're limited to about 100,000 years. Okay? The two basic types of plants that we can get at using carbon isotopes are C3 plants and C4 plants. There's another photosynthetic pathway, the CAM, the crustaceous acid metabolism, but for the most part, pineapples have not been a main food source in human groups. Okay? So we can usually disregard any contribution of succulent plants to the human diet, okay? Unless you're on some tropical island drinking Mai Tais or something. Okay? C3 plants, rice, potatoes, those are some of the biggest crops that are C3 in today's modern world. They're a C3 kind of plant. C4 plants, the biggest one is maize. And they differentiate, they all incorporate carbon through photosynthesis into their structure, but they do it they don't like the heavier carbon isotope. There's two isotopes that we're concerned about, C13, which is slightly heavier, and carbon-12. And this one doesn't like to use the carbon-13. And so it'll make a difference. It results in a difference in the ratio between C13 and C12. And when we plot that, when you run it through a mass spec, it'll count the number of C13s, it'll count the number of 12s, and you get the ratio. When we do that, you can get a chart that looks like this. So here is what's called the delta carbon-13. So it's the number of carbon-13 isotopes to the number of 12 isotopes. And it allows the differentiation based upon the type of plant that an organism may be eating. So a grazer that might be eating more tropical grasses, maize is a grass. It's a tropical grass. And those are generally more commonly C4 plants. Trees, shrubbery, herbs are more commonly C3 plants. So a browser eating that kind of stuff is going to have a C3 signal. These are C3 plants down here. This is where they generally fall out. And these are C4 plants, generally fall out about 0 or a little bit negative. Nitrogen allows us to differentiate between trophic level and the potential inclusion of marine foods. We're concerned about two isotopes for nitrogen, nitrogen 15 to nitrogen 14. So again, we're counting the number of nitrogen 15s, we're counting the number of nitrogen 14s. And what happens with nitrogen is that it becomes, you have a step up in every trophic level. Every move up the trophic level, you have a step up, about +3, +4 parts per mil. And so here's omnivores. Plants would be down here, effectively 0. Omnivores in this case are, from this data, are cave bears. Okay? They're omnivorous. So they're eating most plant material, more than likely, a little bit of animal protein thrown in there. Herbivores are up here. So we have cervids. I don't know what that animal is, just other than an herbivore. There's a bison. And then the trick here is Neanderthals. The plus here is a wolf, and we have a couple of Neanderthals circling around there. So every step up the ladder of the food chain results in an increase in this nitrogen value. So where you fall out will tell me if you're an herbivore, if you're a vegetarian, if you're an omnivore, or if you are on a meat-based diet. And we can do that for everybody in this room. And you fall out somewhere in here. The inclusion of marine food, populations that have a lot of marine food in their diet are higher, even higher than this. Okay? Because nitrogen starts out at a higher concentration. There's more steps in the food chain in the marine environment, right? So we have the phytoplankton being eaten by little fishies being eaten by slightly bigger fishies being eaten by slightly bigger fishies. So there's a lot more steps in the food chain. And so that it leads to a marine diet would be somewhere up here. Right? The other thing is we can also get at what type, if you're a carnivore, we can get at what type of diet your meal ate. What type of diet was your herbivore that you just munched on eating? Right? If it falls out over here, this is using collagen. If it falls out over here, and you're eating an herbivore, that tells me that that herbivore that you were eating ate C3 plants. If it falls out over in this neck of the woods, it tells me that that herbivore that you're eating ate C4 plants. So we have a good degree of resolution possible. In this particular instance, Neanderthals are top carnivores. And they're eating a lot of meat, but they didn't have an exclusively meat diet. There's really interesting evidence coming out that they were eating they have plaque on their teeth. Remember when you go to the dentist and they scrape it with that hook thingy? that medieval torture device, okay? If you don't go and get that done, that stuff can eventually fossilize. It can become mineralized. And it will be a signature of all the stuff that you put into your mouth and didn't brush away. In this instance, they can get DNA, they can get phytoliths, they can get all kinds of information out of this plaque. And a recent publication indicated that there's quite a bit of plant material actually in that plaque. Some of it might be medicinal in nature, but then the other thing that's really interesting is that they might be eating chyme. Anybody know what chyme is? All the anthropology students are going, yes. And they're eating potentially the stomachs and the stomach contents of herbivores. So it's like a pre-mixed Caesar salad. Right? Maybe not too appetizing from our perspective, but a good source of plant material in an environment where plant material is very limited. There's lots of ethnographic modern examples of individuals eating chyme, of eating semi-digested plant material from the stomachs of herbivores. I hear it's a little bit tangy, okay? A little bit sharp. Okay? It's because there's acid in it. So let's get to it. A very brief picture of the last, I don't know how many million years worth of evolutionary history, okay? We have 4 basic groups. The earliest possible hominins, kicking around 6 to 4 million years old. We have the genus Australopithecus. Okay, there's a large number of Australopithecine species. We have paranthropines, and then we have the genus Homo. Okay, so there you go. Let's get, here the earliest possible hominins include, and you don't need to repeat these, Ororhyntugenensis, Sahelanthropus chadensis, and Ardipithecus ramidus. All of them have some features that indicate that they're somehow different than other Miocene apes. Miocene is just the time period, living at the time. They're bipedal. That's really the biggest difference that separates them from any other ape. Okay. There are some dentition and jaw morphological changes that indicate that they're exploiting new habitats, maybe exploiting new foods. Okay. Here's Australopithecus. A couple examples of Australopithecus species, fairly good reconstructions. There's Africanus, there's Afarensis. This is an example of a Paranthropus boisei, okay, with a pretty sweet mustache. I will go more into the differences between boisei and another example called Robustus, and they're really interesting. This is what they are. The different examples, and we'll come back to that. Okay? And then we get into the members of the early members of the genus Homo, where we see some morphological changes that are probably associated with changes in diet. Okay? We have smaller faces and jaws, smaller teeth. We have a small increase in cranial capacity, but there's no change in overall body size or plan. So Homo habilis and Homo rudolfensis are 2 species, the earliest species that we know of in the genus Homo. And then we come to Homo ergaster, or also known as Homo erectus, depending on your point of view. And here we see some significant changes that are probably linked to changes in diet. And what we're really concerned about is this stuff down here, where we see an increase in height. This individual, the Narukatomi boy, was probably about 12 or 13 years of age. And at this time, at full adult height, he probably would have been about 6 feet tall. Okay? A significant increase. This is Australopithecus afarensis, a significant increase from previous Australopithecines. We also see an increase in cranial capacity, but more significantly, we see an increase in what's called encephalization, where we see a bigger brain than expected. Okay? Usually brain size scales with body size, so bigger things generally have bigger brains. So what you have to do is you have to control for body size. And once we control for body size, we start seeing pretty significant increases in cranial capacity. And you have to explain that, okay? And it has to be explained. So just to summarize, Ardipithecus ramidus had pretty big teeth, okay? That's what this says. They had pretty big teeth, okay? But smaller than some of these living primates. Okay? There's no dental microwear data yet. Okay, it's just anecdotal. It's not published yet. This is the stable isotopic data. Okay. Again, this is the C3 end of the range. Right around here would be the borderline, and the stuff over here is C4. So we see a predominance of C3 diet. Maybe as much as 90% of the diet consisted of C3 resources. They're living in an environment that they think is fairly similar to today's environment that chimpanzees are living in, where C3s and C4 plants are everywhere. But they're eating more C3s than they are C4s. The Australopithecines. They also have very big teeth, have larger mandibles, a well-buttressed face, so lots of chewing forces. They have incisors that are as big as should be. Okay? If you fall below this regression line, you have smaller teeth. If you're above it, then your incisors are big. Okay? And their incisors are about what you would expect. But they have big posterior teeth. As a group, for the shearing quotient, they have flat teeth, okay? Relatively flat, blunt molar teeth, lacked the long shearing cusps. Okay? And there's the dental microwear data. Okay? Can you interpret that? Does it look like there's a lot of striations, or is there a lot of complexity? Are there more pits, or are there more striations? Yeah, it looks like pits to me. So here's Australopithecus africanus, the open circles. And so they're falling over here towards the tough end of the food spectrum. Right. And there's stable isotopic data for two different species. Bahar al-Ghazali, which indicates somewhere between 50 and 80% of their diet may be C4. Here's Australopithecus africanus, so this is really across the board. A fairly even split between C3 and C4. And what's really interesting is the range of variation. And then the paranthropines. The two that have been studied for stable isotopes and dental microwear are robustus, sometimes called the nutcracker man. So you can kind of think about what people thought that these individuals were eating. And then boisei, which sometimes has been called hyper-robust. So all the cranial dental morphologies of these things indicate that they're chewing lots of hard things. Okay? Lots of chewing forces. Sagittal crest, big cheekbones, flaring face. Okay? But when you look at the dental microwear data, there's a divergence. We have boisei here in the filled-in circles at the tough end of the range of microwear, and then robustus is down here with little orientation, but a lot of complexity. So they're eating, despite the fact that they look the same, they're eating distinctly different things. Okay. Robustus probably consume more hard and brittle, okay, over here. And boisei was not a hard object feeder. They weren't eating nuts. Okay. The stable isotopic data is very interesting because boisei, has one of the highest C4 diets of anything that has ever been studied. Okay, outside of one thing that I'll show you in just a sec. Robustus is down here, a little more C3 than C4. Boisei has a C4 signature that is very similar to this type of gelada baboon. And as fierce as these things look, they graze. They like whole hundreds of individuals in a herd, and they're just grazing along the savanna. And what they do, they're picking up corms. They're taking the grass, they're pulling it up, and they're eating those little bulbs underneath. Okay, the little underground storage organs. So these are underground storage organs. This is where the plants keep their energy. Okay, they're a carbohydrate, they're storage form of carbohydrate. And so it's a good food source. And so the suggestion is that they may have been eating, they were grazers, that they were eating these C4 corms of grasses. Whereas this thing was probably eating a more traditional hominoid diet of a mixture of fruit and foliage. What this does is it calls into question the monophyly. Do these things actually have a common ancestor, a recent common ancestor, or are they paraphyletic, where we have convergent evolution of this unique cranial dental morphology? I don't have the answer for that. It also highlights the dietary adaptation paradox. Both of these things look like they should be munching on acorns or walnuts or something, but they aren't. This one, fruit, maybe some leaves. and this one seems to be eating corms, underground storage organs of C4 plants. Then we get to ergaster. A summary of ergaster. A lot of interesting things happen with Homo ergaster. And so we have thicker tooth enamel, a pretty robust mandible, more occlusal relief on the teeth. They're using tools, okay, we're going to come back to that. Unremarkable dental wear. And that's not my words, that's the words of the author. And then they think their stable isotopes indicate 60 to 70% C3 plant use. But the most interesting thing about Homo ergaster, which I already mentioned, is that we start seeing an increase in encephalization. We start seeing an increase in cranial capacity greater than expected based upon increase in body size. Right here. Okay. This is an cranial capacity, which is a rough measure of encephalization. Here's Australopithecines that we were just talking about. Here's Homo habilis, Homo rudolfensis, and here's Homo ergaster, there's Homo erectus. We see two periods in hominin history of relatively significant increases, bigger increases in cranial capacity and of encephalization. The first one is here with Homo ergaster, Homo erectus. The second one is right up here with archaic Homo sapiens. Okay? And the question is, how? Brains are a very expensive organ, so don't let them go to waste. Okay? They're very, metabolically, they're very expensive. They contribute, they make up about 2% of your body weight, but they take up about 20 to 25% of your daily caloric requirements. So there's a lot of things going on in there most of the time that requires energy. And so we have to find that energy from somewhere else. Your guts are also a very expensive tissue. Okay? And so there's a very interesting thing that potentially is happening where we see a reduction in the size of the gut and an increase in the size of the brain. But that had to be facilitated or at least grounded on a change in diet. And there are two different ideas regarding that change. One is cooking. And if you go to the Evolutionary Studies website, there's a talk by Richard Wrangham, who's the most articulate architect of the cooking hypothesis that suggests that around 2 million years ago, hominin ancestors started cooking food. Not necessarily meat, but potentially corms, underground storage organs, tubers. Unfortunately, it's hard to evaluate that because the oldest evidence of controlled use of fire is about 400,000 years old. And so it's a million years off. We can't even tell. Okay? But there is very good evidence for meat. Okay? For meat consumption in the form of faunal assemblages, of broken up, of cut human or anthropogenic cut marks on bone. These are just two sites from where such material comes from. This is very old. Okay? This is very old and is weird. Okay? Because we don't expect tool use this old, at 3.39 million years old. We don't expect that. That's pre-Homo. And we don't think that there were tools. The oldest stone tool is at 2.6 million years old. So how this happened is very, very interesting. And people much smarter than me look at this stuff and they can differentiate between a mark from a stone tool from a mark based upon grit that's in the ground that has been encased for the last 4 million years. Right? And they can tell the difference between a cut mark and a canine tooth mark. And they can tell the difference between a percussion mark where you bang it with a round rock to get at the bone marrow, good eating, okay, and where you're cutting off ligaments and tendons to get at the muscle mass. And this is what these indicate. What it indicates that we're having primary access to meat, not necessarily scavenged. We're not necessarily a secondary scavenger or a passive scavenger. We may be a power scavenger at this point in time, where we're actually getting either to kills, maybe not making the kills, but we're getting the kills very early, and we're able to scare off whatever predator may have actually killed the thing. And so we're getting access to big hunks of meat, okay, rather than just the dribbles that are left after a lion goes through a kill. If it's the dribbles, then you'll see percussion marks where we're getting into the bone marrow. Okay? If we get primary access, you see a lot of cut marks because we're taking the big chunks of muscle. So we see this occurring. We know this is occurring. And it wouldn't take a lot of meat being included into the diet to have a significant impact. in human evolution. So, I'm not going to talk about Neanderthals. We're stopping at basically 2 million years ago, and we're going to jump up to 10,000 years ago. There's a whole interesting time range in there that has all kinds of data in it, but just don't have the time. Okay? But there's lots of studies out there that look at the difference between Homo sapien diet that are living contemporaneous with Neanderthals and differences in the diet that may indicate differences in foraging strategy. And one article that I know about that I recall well enough is that modern humans may have been able to access rabbits a little bit better. That they might have had the technological sophistication and adaptability to get at fast-moving, ubiquitous prey like a rabbit, whereas Neanderthals were principally big game, and that may have limited their access to resources in some way. So there's all kinds of information out there. But we're going to talk about the Neolithic Revolution. Okay? This map depicts centers of independent evolution or appearance of food production. Independent. So these are primary centers. This is not where eventually agriculture diffused into other world areas. Okay? But this is where it originally developed in these specific areas. Okay? The oldest evidence is for the Levant. Okay? In this neck of the woods, in the Fertile Crescent, Iran, Iraq, around, this says 10,000 BP. The present is 1950, so it's about 12,000 years old. And there's evidence there. It's right around that that's right around that time frame. And we're seeing the beginnings of plant domestication and beginnings of animal domestication occurring almost at the same time. Okay? The traditional picture is that plant domestication happened first, and then animal domestication. But the evidence is now starting to bring those two much closer together. And there's all kinds of data out there, all kinds of research that's looking at morphological or using morphological data of these plants, of these animals, to identify this is domesticated and this is wild, and looking at that transition. We're going to look at some of the biological consequences to humans. There are biological consequences to plants and animals, but they're just food, so we won't worry about them. We're going to look at, very briefly, I already kind of mentioned this during the pre-talk, these are 3 different categories of change that accompanied the shift in subsistence. The first one is craniofacial changes. With agriculture, with food production, we also get the almost simultaneous invention of ceramics, of fired pottery, which is able to withstand repeated firing and cooling, or heating and cooling, so you can cook your food. With that, we see a significant change in the texture and the nature of the food itself. We reduce the amount of masticatory effort that is required. Our diet becomes softer. So that's going to relax the natural selective pressures that are maintaining a thick mandible, big teeth, strong attachments, and so our face becomes more gracile, becomes smaller. It changes, and it changes faster than our teeth change in size. So our jaw and our maxillae get smaller while our teeth still kind of stay the same. So we see a lot of dental malocclusions occurring in that transition from food foraging to food production. Activity patterns change. We can reconstruct activity patterns based upon cross-sectional geometry of bones, because bones respond to stress. And so when we can evaluate the strength of your bone at particular segments of the bone, we can say, oh, it's stronger in this plane than in this plane. And knowing where the muscle attachments are, we can reconstruct to a degree the type of activity and the load of activity that people are engaged in. And we see significant shifts in bone strength from food foraging to food producing. Overall, we see a decrease in bone strength from food foraging to food production. Finally, the last category is health and disease. The pattern of disease in food-producing groups is largely attributable to changes in population size, ecological changes, and then the nature of the diet itself. So we'll go through those quickly. We're actually going to come back to this, so I'm going to hold off talking about that for a minute. But the larger population exposes us to new disease vectors, new pathogens, nasty little microscopic parasites that are due to contaminated water. Living in close proximity, we make trash. okay, of all sorts. And so with an ineffective trash disposal system, it creates environments that these little guys like, and it leads to diseases. Okay? We also are exposed to new pathogens. Once we start domesticating animals, we're going to be exposed to their pathogens. Living in close proximity with bovines probably resulted in us getting tuberculosis. The closest genetic relative to our tuberculosis is the bovine form. And so it jumps ship. These things are able to evolve quickly, to adapt quickly to new environments, and that's likely what happened. This is a view I had outside my window for a summer, living with goats. So I have no idea what I've been exposed to. The nature of the diet itself. Okay, so that was ecological changes. The nature of the diet itself, for the most part, food-producing groups have a less nutritious and a narrower diet. Food foraging groups generally have a very wide set of foods that they consider to be food, like chyme. Okay? We generally have a much more restricted conception of what food is, or what is appropriate food. And in large instance, in many examples, that food is less nutritious. In this particular example, we're talking about maize. If you have a predominantly maize diet, it lacks some essential amino acids, or they're chemically bound and you can't get to them. Essential amino acids means that you have to get it from your diet. You cannot make it. If you have a predominantly maize diet, the vitamin B3, which is necessary for catabolism of these things, is chemically bound. You can't get to it. Okay, it's there, but it's bound up. And it leads to what's called pellagra. Okay? It's a result of the improper synthesis of collagen, okay, which is ubiquitous in you. And eventually, if you do not have vitamin B3 or niacin, you will die. And indigenous groups that subsisted on maize had figured this out a long time ago, and they have cultural means of adapting to this. Anybody hear of hominy? Anybody know what hominy is? It's nixtamalized. You put basically, what is it? It could be an ash, it could be lime, and it allows the release of this niacin. Tortillas are the same kind of process, making tortillas. But if you don't, you can get pellagra and die. So a single monocrop diet can lead to some fairly severe health consequences. Okay? Skeletal manifestations that we would look at to evaluate this include osteoporosis, and I'll go through these quickly. This is a bone infection. Okay, so this is a bone infection in a living individual, living in here as well, but that's how it manifests. Okay. Dental health. This is a cavity, okay, a very large cavity. Every time I see these pictures, I always want to go home and brush my teeth. These are what are called, these are enamel defects. Okay. Your enamel deposition is very sensitive to environmental disturbances. And so this is a very severe environmental deficiency. Okay, something happened, this individual probably had some kind of pretty bad disease and during early childhood, and it resulted in the very poor enamel development. Iron deficiency, nutritional deficiencies can manifest skeletally. This is called parodic hyperostosis, and this is called cribra orbitalia. Your skull, the area between the outside and the inside, it's called the diploe. And it's a site of hemopoiesis. It's where red blood cells are produced. A lot of red blood cells are produced there. And in iron-deficient anemia, there's not enough iron. And your body for the red blood cells to transport oxygen and carbon dioxide. And so the body tries to respond to that. And it proliferates these tissues. And it will expand. This is an expansion into your orbit. Okay, so more than likely there's some kind of soft tissue manifestation as well. This is the general picture of what happened from food foraging to food production. Okay? Infection goes from low to high, dental caries goes from low to high, enamel defects goes from low to high. You kind of get the picture. Okay? For the most part. Not every population is going to go from low to high in these indicators. Some won't change at all. Okay? So the general picture is this way. And the question becomes, why would anyone do this to themselves? Okay? Why would any population decide upon and follow through on this course of action? And how we have to evaluate that is the final part my final point is distinguishing between physiological fitness and Darwinian fitness. And I don't think I gave the classes these articles, but if you're interested in this, I can send it to your instructors. Physiological fitness is probably the way that we normally think about when we hear fitness. Okay, hey, you look pretty fit. Thanks. You know, it's mostly physical attributes. This is really homeostasis. Okay, physiological equilibrium, not being sick. Darwinian fitness is reproductive fitness, the ability to both survive and reproduce, and your average contribution to the next generation. So these are all the bad things, okay, that happen in the transition from food foraging to food production. And the question is, why in the world would anybody do this? Okay. Some would say that they can't actually foresee these negative consequences, and that's legitimate. Okay. But why wouldn't we go back? And the question, the two things. One perception is that we were pushed into agriculture. We had no choice. Once you start making more food, you're going to get more people. More people mean you have to make more food, and so you have this feedback mechanism where you need to intensify your food production. So we were pushed into agriculture because of increasing population. The flip side is that we were pulled into agriculture through natural selection. There's this concept called niche construction. All organisms modify their environment in some way to increase the chances of survival. Beavers make a dam. We wear clothing. Okay. We had agriculture. So in this way, agricultural production could be conceived of as a way produce more food, and in combination with development of pottery may have facilitated an increase in fertility. From an evolutionary perspective, agriculture is good because it increases fertility. There's more food, high carbohydrates, high calorie, which is good. Okay, lots of energy. So instead of being pushed into agriculture because we had to, because our population is increasing, maybe we were pulled into agriculture because natural selection is favoring the construction of a particular economic niche. In this way, in this form, agriculture. So instead of saying that agriculture is bad, 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 and it is, from a physiological fitness point of view, from an evolutionary perspective, from Darwinian fitness perspective, agriculture has been phenomenally successful. How many people are there right now? 7 billion? From an evolutionary Darwinian fitness perspective, agriculture has been phenomenally successful as an adaptation. So we can't look at it from one perspective. We have to combine the perspectives in order to understand the evolutionary significance of the human diet more fully. And I meant to have a picture of sheet cake and pizza, but I couldn't find any on the website.
We will have some soon.
Yes. So if you want highly processed sheet cake with lots of carbohydrates and lots of calories, there will be some in the terrace. Thank you. Yes, Dr. Mass.
So, Ken, you were talking about
Yeah, I didn't put this in there. The paleo-environmental reconstruction indicates that Boise, the darker one, was living in a little wetter environment where with sedges. They think they were eating sedges, potentially, like papyrus. It's a form of grass or a rush. And the other one was a little bit more savanna-like in terms of its paleo-environment. So they're living in different areas, which is still interesting because they look almost exactly the same in terms of the morphology. Yeah, yeah. Those two, the Ardipithecus is the common ancestor between those two. So those two are not directly ancestor-descendant relationship. Boisei and robustus, they're a different.
No, I'm talking like overall.
If there's retention of juvenile characteristics in Paranthropus, hominins. Okay. I guess in some sense there is the greater retention of neotenic features because we generally tend to have a higher forehead, a bigger forehead, and a larger, more globular head, which is similar to our infants. But in terms of those, they didn't have a forehead, so I can't really speak to that too much. Because that's not my field, really, so I can't really address that too well. But thank you for asking a question that I couldn't answer. Shannon? I think it would be very difficult in today's world to subsist on a foraging diet in the Western world. It's even getting more difficult to do for groups that are even remnant for food foragers because they're being pushed to increasingly marginalized environments. And so the environment can't sustain it anymore. They're being pressured by encroaching agriculturalists or capitalists, and so the resource base isn't there to actually maintain a food foraging lifestyle anymore. I think that it is, in terms, the paleo diet phenomena, I mean, it addresses the health consequences of that type of diet, and the health benefits of switching to that type of diet seem to be, I mean, it's good. It's good to switch to that type of diet. It's good to avoid highly processed food. It's good to avoid lots of sugars. It's good to avoid hormones. It's good to avoid all the bad things that are put into our food. There's no doubt about that. So if you switch your diet, you're going to improve your health. But when we talk about it from a broader scope, a broader perspective in terms of adaptiveness and understanding the difference between fitnesses and between the fitnesses, we have to appreciate the understanding that there's this other aspect to it, that agriculture, despite its negatives, and a lot of the negatives that are along with our modern Western diet are much more recent than what I was talking about here, okay? The introduction of beet sugar, living in megacities, all the accompanying stresses that impact your health that are unrelated to food are going to negatively impact your health as well. So I wouldn't say it's I wouldn't go so far as to say it's a cop-out, but we need to understand, appreciate both sides of that spectrum. Okay? I certainly don't do paleo diet. I like too many bad foods. And yeah. How does pottery? Because you're able to cook food. And so you're gonna be able to cook more food, and the carbohydrates will be converted. They're gonna be broken down a little bit more, and so digestion will actually be a little bit easier. And so you're gonna be able to make weaning food. Okay? You're gonna be able to make gruel for your baby, which means that you can switch them off of breastfeeding to solid food earlier, which means that a female can come into ovulation again and have another child, while the other one technically is still nutritionally dependent upon the mother. Everybody get that? Okay. Yeah. Uh-huh. Well, I didn't choose it, for one, but I don't know why the bears were lower than the herbivores. I don't know why the cave bears were lower. They might have, I'm not sure why the bears are lower. It's interesting, but I can't speak to it right now.
Usually they should.
You would think if they're having any kind of animal protein in their diet, they should be higher than the herbivores. Yeah. Yeah, grasses. I suppose it might depend on if it's a nitrogen fixer, if the plant is a nitrogen fixer as opposed to one that is not. I can't remember if they've done any controlled feeding studies if they're just eating legumes versus just regular old, I mean not regular old plants, but just non-fixating ones. But that could potentially explain if they're eating fruits. You know, I don't know. I can look. If If anyone's interested, you can email me, and I can actually send you the article, and I can try to shed some light on that. Yeah.
I know you don't work on this, but do you learn about ancestral diets by what nutritional requirements appear to be behind
Right.
it? So, like iodine, augmented nutrients,
Mm.
then tell us something about our ancestral diets.
Well, assuming that our ancestors also needed iodine, then looking at our nutritional requirements in modern groups should inform, assuming biological uniformitarianism. If there's no change, then I suppose you can assume that. But there is, I mean, there can be changes that would not make that a one-to-one. I know that there's been a lot of looking at much more modern groups that are much more akin to food foraging groups, looking at their nutritional intake and the differences, the vast differences has between us and them, modern Western diet versus them, which is much of the basis of the paleo diet, correct? I mean, when I read Cordain and stuff like that, they do a lot of ethnographic analogy. Enjoy in moderation. No, really. I mean, that's really it. The problems with our Western diet is that it's skewed. Okay, we have ready access to all the wonderful tasting but bad for us food that we can never want. But if it's in excess, that's where things start breaking down. We have an amazing plastic physiology, an amazing phenotype that allows us to accommodate an incredible range of dietary intake. But if you get skewed to any way, then you're going to start having negative consequences. So enjoy the meat, but don't solely subsist on the meat. Even the Inuit, with a predominantly meat-based diet, incorporate plant material in some way, shape, or form. Anything else? I'll be teaching this class again, so if you're interested, look for it, and thank you.